Jamie, it's nice to see you, and I believe you're going to tell us about the project you've been working on here at the Roland.
Yes, so we're really interested in behavioral variation in general, and with this project, we're specifically interested in variation behavior between individuals. So how does one fly differ from another fly, even if they're very closely related or identically related? If we think back to humans, we know that every one of us has a unique personality, and dog and cat owners will agree that even their pets have unique personalities. And we think this is true with fruit flies as well. And so we're interested in studying that and seeing if we can come up with the mechanisms that generate these unique personalities.
You're going to be telling us about a technique you developed for your project here.
So the device we built and that we'll look at is called FlyVac. And the vac will become obvious because it involves using a vacuum. But so essentially we're going to study this behavior called phototaxis. So if you shine a light, fruit flies, like a lot of insects, are generally attracted to the light and will move towards it. That's an oversimplification. Like if you study, if you look at a group of flies, even if they're identical genetically, you're going to get a range of responses to light. And so the old way of studying behavior, when you're just interested in studying phototaxis in general, you just measure a bunch of flies all at once and see kind of how the group average responds to light. And what we were interested in is how does each animal respond to light and how does that compare to the others? We're more interested in like how wide is this distribution? How far do the tails go out? And so using this device, we can screen many thousands or tens of thousands of individual flies and get a behavior score for each one.
So what does being able to test the behavior of thousands of flies at a time get you that you wouldn't be able to do if you didn't have this high-throughput device?
Well, in order to study individual-to-individual variation, it's really important to be able to assay many thousands or tens of thousands of animals. And that allows us to easily find individuals with kind of, extreme personalities, like they really like light or they really don't like light.
Would you agree that at the moment we don't know how to explain why one animal might really like the light, but the next animal doesn't?
The genes and neurons and whatever it is that makes the fly have this unique personality, we don't know the answer yet. This device is to show that flies have unique personalities and that these personalities persist. So if we find flies that are extremely light-loving or extremely dark-loving, and we can measure the fly and then save that fly and measure them a day later or a week later or a month later, which is really the lifetime of a fly. And we find there's a persistence in their personality. So whatever it is that makes them the way they are, it lasts for their whole lifetime.
So I see you have one of the modules from FlyVac.
Essentially, there's 32 of these attached to one big device, and so each one will have one fly. So you measure 32 flies at once, but each fly is being looked at individually. Basically, each one of those devices is a maze. It's called a T-maze, which is kind of a classic maze for behavior. It's very simple, and that's why it's very powerful. So the fly is going to start here at the bottom, and will climb up this maze to a choice point. And so when it gets to the middle here, there's going to be two light sources, and one is on and one is off, and that's at random, so it can switch back and forth. And so when the fly gets there, if he likes the light, he's going to turn and go towards it, or if he doesn't like the light, he'll turn the other way. And we have some infrared beams at both choice points, so that'll detect the fly's decision, and the computer will record it. So if this fly goes up and chooses light, he crosses that beam, this fly chose light, and then the important thing that our device does, and here's where the vacuum part comes in, is there's a vacuum here. So once the fly makes a choice, the computer actually turns on the vacuum, which sucks the fly back to the beginning. And so this is very important because if the fly only did this once, it wouldn't be that useful. This way we can do this test many, many, many times. In fact, we usually do 40 times, somewhat arbitrarily. And we can get a really good measurement of what the fly's preference is. And so maybe this fly chooses light, 30 out of 40 times, but maybe the neighboring fly chooses light 10 out of 40 times, or things like that.
You said that most flies like to go toward the light. How much does this vary within a strain of flies, and how much does it vary between different strains?
Yeah, most flies like to go to the light. We've looked at, I don't know, maybe 30 strains. And so you do find variation. And obviously, there are some strains that are blind, and we test those as controls. And the blind flies, turns out, not surprisingly, they choose light 50% of the time, and there's very little variation because, as you would guess, you can't find extreme light-preferring or light-hating flies that are blind because they don't know. But if you're looking at normal strains, all the strains show surprising variation. So no matter what strain you're looking at, you can find these individuals that are very light-loving or light-fearing. But some strains have more variation than other strains, and so that suggests there's a genetic basis for how much variation a particular strain shows.
Does this mean that in lines which exhibit high behavioral variation, you find high genetic variation?
Surprisingly, no. So we've looked at, so flies, you can inbreed strains and get them to be basically isogenic, meaning there is no genetic variation in them anymore. So a particular strain that's inbred, you can measure 1,000 individuals that are all genetically the same, and you still see a lot of variation. What was surprising is we went and caught wild strains. You might expect that a strain that has a lot of genetic variation would show more behavioral variation than these inbred lines. We actually found the opposite. So it turns out the more inbred they were, the more behavioral variation they showed.
So I understand that inbreeding is often insufficient to make a line isogenic. You can have inversions and you can have allelic combinations that are lethal, and ultimately you can get parts of a whole chromosome that are still heterozygous. So was there another more definitive test you could do to show that it's not cryptic genetic variability in these lines that explains the behavioral variability?
Yeah, the big test we did is if you take an inbred strain and you measure its behavior, and you find this distribution of behaviors, and you have these extreme behaviors on the ends of these distributions. So you have really photopositive and really photonegative flies. You can take those guys, find a male and a female that are really extreme, and if you mate them together, they'll give you a whole new population of flies. And you would expect if the reason that they're really photopositive is because they have some genetic allele for that, then it's going to be greatly enriched in their progeny, and you expect the whole distribution of behaviors to shift towards the light. But what we find is when we do that experiment, if we take the extremes and mate them and get the next generation, those flies actually have the exact same distribution of behaviors as the original parents.
So if genetic differences between individuals can't explain their behavioral differences, do you think there exist any genetic differences that explain perhaps the extent of variability within a strain?
Yeah, so we looked at many strains using this device, and some strains have a lot more variation than other strains. The strains that had a lot of variation often had this mutation in the white gene. It was kind of surprising at first because the white gene is mainly known for its role in the eye pigment. So fruit flies normally have red eyes, but if you have this mutation in this gene, you have a white eye. But it still wasn't really clear why having this mutation would cause just generally variation in their phototaxis behavior. One theory for that is that this gene actually has kind of a wider role. It might be involved in neurotransmitter synthesis. So there are some reports in the literature that it could be important for synthesizing dopamine and serotonin, which are two major neurotransmitters in the fly brain and in people. So they're very evolutionary conserved neurotransmitters. So we kind of suspect that there's neurons involved in controlling the magnitude of variation.
Have you done any experiments to distinguish the role of serotonin from the role of dopamine?
Essentially, using drugs in the food, you can increase or decrease the level of dopamine or serotonin. And so in doing that experiment, we found that serotonin, but not dopamine, will control the magnitude of variation. So if you have a strain with this white mutation, it normally has a lot of variation. And we think maybe that's because it has low levels of serotonin. So if we feed them serotonin, we can actually decrease the variation to kind of a wild-type level. Likewise, if you have a wild-type strain that normally has less variation, and you can give them inhibitors of the serotonin synthesis pathway, so that has the effect of decreasing serotonin levels, and that'll then increase the variation in the wild-type strains.
Are there follow-up projects that you're considering pursuing?
Drosophila is really useful for mapping neurons, and there's a lot of techniques for that. So it'd be really neat if we could find, the specific set of neurons. There's probably just a handful of them, where if you turn them off or turn them on, you can change the amount of variation you see in a strain. We'd also like to study behavioral variation in other behaviors. We think, we've been talking about phototaxis this whole time, but we think these behavior, this personality and behavior, we think it's true for all behaviors.
What would you say is the ecological significance to this idiosyncrasy in behavior, behaving differently from one another?
Right, that's a big open question. Is why is there personality in behaviors? Like if it's good for flies to go to the light, maybe they should always go to the light, and there shouldn't be variation. And so we think there's a couple reasons that we might see this. And so maybe it's actually bad to have this variation, but it's too energetically costly. Maybe you could do it, but you would need more genes and more energy and more neurons to have a really precise response every time. But it could be adaptive. Most of the time, maybe it's good to do a certain behavior. But then, maybe there's a drought, and that changes what the ideal behavior is. And so for that, that rare drought, it's better to have the alternative behavior. And those flies will survive. But then the drought is over, and we're back to normal conditions when the other behavior is better. And those flies that survive the drought, since it's not genetically heritable variation, their progeny will have the original distribution of behaviors that's well suited for a normal environment.
So it seems like adding randomness to behavior, which may seem maladaptive, could actually be beneficial when it's integrated over a variety of selective pressures.
That's right. That's what we think.
Okay. Let's go take a look at flyback and see how it works.
Yep, sounds good.
Okay, so here we are back at the lab. And what are you working on, Jamie?
Here I'm getting flies ready to be assayed. So I'm just separating males from females. This way, to the flyback.